Let's start here again with the transfer RNA molecule. So it's this clover leaf shaped molecule that is made out of RNA. It's a short stretch of RNAs, sometimes double-stranded, partially double-stranded, and partially single-stranded. It carries on one end an anticodon that has to match a code word by base pairing rules. Has to match the mRNA triplet that we're reading at the ribosome, and then it will deliver on its other end. It is sort of linked to an amino acid that is specified by the anticodon right there. So the amino acid, you can always look that up by looking up the mRNA code word that then that specific transfer RNA would have to carry. And again, so the transfer RNA carries an anticodon that matches the codon by base pairing rules on the mRNA, and then carries the amino acid that is specified by the codon, the mRNA codon. Okay, so and then the ribosome will read triplet by triplet, wait for the appropriate anticodon to come around and deliver the next amino acid, and then these amino acids, they will be attached to each other by peptide bonds. So basically the routine is that the mRNA codon will be read by the ribosome. We need the anticodon of the tRNA to come by, briefly bind to that mRNA codon, and then deliver the amino acid that is specified by the mRNA codon. And then the discharged tRNA molecule will be leaving, and the only thing that needs to be done is that the two amino acids need to be brought in close proximity, and then we need to make a peptide bond between two amino acids, and that will cause the polypeptide chain to grow. Okay, so here one more time, the translation of the mRNA. This is a very crowded slide. It's sort of the all-inclusive, has it all slide here. But so here we have the messenger RNA. Let's start over here. And we're reading these code words. Here's code word A, B, C, D, and so forth. These are all triplet codes. And the ribosome is here in red. And you can see that we're currently reading triplet D, code word, this triplet D. And the tRNA molecule is there, and it will deliver the fourth amino acid. Then we're going to move on. We're going to read triplet E, and we need to wait for the appropriate anticodon on tRNA number 5 to come. You have it bigger over here. D and E, these are the two in holding position at the ribosome right now. Then once we have these two tRNAs with the specific amino acids that they deliver, then we just need to form a peptide bond between these amino acids, and then the tRNAs will be discharged. They will be leaving, and then we need to get the next one to come along. So here we have 4 and 5 are now connected. Now we need to put the reading frame one triplet over in the ribosome, wait for the next tRNA that matches that codon that we have in hold here, and then deliver the next amino acid, link it up 5 to 6, will be linked up by the next peptide bond. Okay, that is translation. So here one more time, in this very beautiful mechanism. So this is how all the proteins in your cells are being made. You have the small subunit and large subunit of the ribosome right here. Right now we're holding here, you always hold 2 triplets at the ribosome because you need to make sure that the peptide bond forms. So we have here triplet GCA needs to wait for anticodon CGU on a tRNA, which delivers an alanine amino acid. Then we have it also on hold here, UCC, and that is the anticodon AGG. And now we need to wait for the appropriate tRNA to come by, and that one actually delivers the serine amino acid, and we just need to form a peptide bond between these two, and then the 2 tRNAs can be discharged. And here comes already the next one. Okay, so here's another aspect of this Usually an mRNA has a short half-life of, let's say, 20 minutes or so, 30 minutes maybe. So you want to make sure that you don't just create one protein product, but several. And so right away you basically get several of these ribosomes to work. So they're in gold here, these ribosomes, so we call those polyribosomes. It's like, picture like workers on an assembly line. They are reading through the mRNAs, several of these ribosomes. They will attach to a messenger RNA molecule, and they all make the same protein product. And the mRNA, as I said, it doesn't last very long. It's only about a 20-minute or so. Okay. Once we have that primary sequence of this polypeptide chain, so the amino acid sequence, the protein product isn't finished. And so the alpha helices and beta pleated sheets, the secondary structures, they usually form automatically just by attraction within the molecule itself. But then the tertiary and also any other attachments or even quaternary structures, they need to be formed at the level of the ER and the Golgi complex. So if we need the ER and the Golgi complex to really work with that protein and make it, usually it's also being cut, attachments are being made, it's being folded and shaped into a functional protein at the level of the ER and the Golgi apparatus. So these proteins that are being made, these ribosomes, remember that we have rough ER, the ribosomes are sitting literally on top of the ER, and they will be secreting or sequestering this brand new polypeptide chain into the interior of the ER. And then the interior of the ER will take over from there and shape the protein into what it needs to be to make sure that it's a functioning protein with the appropriate shape. So here is a nice picture illustrating what I just said. So here are your ribosomes in red, and they're reading the mRNA, and there's this growing polypeptide chain, and right away this polypeptide chain is being sort of secreted into the interior of the ER. So here we're putting this polypeptide chain, and then we're going to add carbohydrates. Maybe we're going to fold this appropriately, shape it up to its three-dimensional shape, and whatever needs to be done. It's different for every different type of protein. And then off we go. We're going to package this up into a vesicle and send it off to the Golgi apparatus, which then decides on the final destination of this protein product. So here, sort of the sequence of events of what can happen to different types of proteins. So we have two different scenarios here. The scenario here over to the left would be that, so we have here our messenger RNA. It's being read by the ribosomes. We're making these protein products and putting them into the interior of the ER, and it's being shaped and altered and folded and whatnot, then packaged up into a vesicle, handed over to the Golgi apparatus here in purple, and that one will do its final touches on it. And then here we're packaging up the final product, and by exocytosis we're going to let it out of the cell. So this could be, let's say, I don't know, this could be a human beta, it could be a pancreatic beta cell, and it's making insulin, and now we're packaging up the insulin, and we're just going to dump it out into the blood. Okay. Scenario B over here on the right, that is a protein product that the cell needs. So same procedure. Initially we're going to put it into the ER for folding purposes, hand it over to the Golgi, but then the Golgi just takes it, packages it up, and moves it wherever it needs to go in the cell. So in the case of protein B, was apparently that's something that was supposed to go into a lysosome, some protein, and so we're going to put it into the lysosome, and then the cell will make some use of it. Okay, so now we're done with transcription and translation. So the purpose of transcription and translation was protein synthesis. So clearly we need the mRNA as a copy of the instructions that we found on the DNA, and then the mRNA then works together with the ribosome to make a protein product, which then needs to be processed and folded and whatnot and shipped wherever it needs to go, and then fulfill its function. So clearly transcription and translation, the purpose is protein synthesis. Now let's move on to DNA replication. The purpose of DNA replication is to make a copy of the entire genome, DNA to DNA. So make a second DNA copy, the entire instructions, everything that the cell has, that organism is specified by in its genetic material, everything needs to be copied, start to finish, in preparation of cell division. So in a nutshell, DNA replication is making a copy of the entire genome in preparation for cell division. And here the product is another DNA molecule. So we're going DNA to DNA and the base pairing rule. And let's take a look at how this works. So this slide here actually explains it sort of in a nutshell, what replication is all about. So you have a double strand of DNA. Each one is a template strand and serves as the template for a secondary DNA strand. So we basically have to open up the double strand of DNA, and because eukaryotic genomes are really large, you have these multiple origins of replication. So these replication bubbles, they open up multiple places, and then basically you're synthesizing a secondary strand based on base pairing rules, and then you're fusing these bubbles. Eventually they fuse together, and so you're going to end up with two DNA molecules that are identical, except that we call this semi-conservative. One strand of each new DNA molecule is an old one, the blue one, and the gray one is the new one. So, but both of these DNA molecules are identical to one another. DNA replication is extremely efficient and extremely high fidelity. Even a genome of the size of a human cell, so human genome is 3 billion base pairs. On average, there are only 3 mistakes made. There's a lot of proofreading function in replication, and cells are extremely careful to not make any errors. So think about it. The genome, human genome, 3 billion base pairs, or think of them as letters. That is 3,000 textbooks, which have 1,000 pages each and 1,000 letters on each page. And you're supposed to copy that error-free. It's pretty good what cells can do if they only introduce like 3 mutations, 3 errors per replication cycle. That's extremely good. Anyway, so let's take a little closer look at replication, how it works. So basically, this is a replication bubble that was cut open, so it would continue here in blue and down here in blue. So but we cut it open basically, and so you see only half of it. You start at the origin of replication, and so we open up this double strand right here, and there's one thing about DNA polymerase that's kind of odd, and that is that it can only go in 5' to 3' directions. So on the leading strand, what we call the leading strand here, this is not a problem because we're starting here. So opposing a 3' end on this coding strand we're starting with the 5' end of a DNA molecule, and we'll start running down here. Using the base pairing rules, here's your DNA polymerase molecule. So this is the enzyme that does the job. And it's zooming down here, attaching nucleotides based on the base pairing rules, all the way until we reach the end of this parental DNA strand. So no problem here, because we're going in 5' to 3' direction, okay? Now, on the opposite end, the opposite strand here, the second coding strand here, or parent strand, the problem is we also can only go in 5' to 3' direction. And let me get a different color here to make that clear. So in other words, we cannot go in this direction. We can only go in this direction here. And because that is the case, we have to put down a little primer that's called an Okazaki fragment, so a little primer piece that's made out of RNA. And then we have a starting point, and now we can go in this direction, in 5' to 3' direction, so we can finish this piece. And then we need to wait for the next stretch of the DNA to open up for us, we need to put down another primer piece right here. And then our DNA polymerase can go in its favorite direction, which is 5' to 3'. So we're going to go this way. And then we have to wait for the next one, and that starts right here, and we're going in this direction. So I think you get the point. So then here we have to wait for the next section of DNA to open up. We need to put down another primer, and then we can finish this piece. So we have to, in other words, we have to synthesize the lagging strand in pieces, and obviously that takes longer. Therefore, this is called the lagging strand. Okay, so also, the additional step is we need to remove the primer and replace it with DNA nucleotides, because the primer is always RNA, because it can start any place basically. So there's a lot of stuff to it. To be honest, I had to take a whole course on replication alone. It's not so simple, but sort of trying to understand sort of the principle of it, this is good enough. So it's not as easy as what it looks, and you might even think, oh wait, already the lagging strand seems a little complicated, but there's a lot more to it actually. So anyway, trying to keep it sort of manageable here. The leading strand is not a problem, so we're going in this direction up here. That's not a problem, 5' to 3', but the other side here has to be synthesized in pieces. And let me just demonstrate this here as a continuation. Of course, if you're starting here on this strand, you can go, this is now your leading strand, this is your leading strand going in 5' to 3' direction, the way it wants to go, and go all the way to the end of that chromosome in this case. Now up here, now we have the problem because we also want to go in 5' to 3' direction, so we're going to have to do the same thing. We're going to have to start to put down a little primer piece right here, and then we can go in this direction. And then we have to put another primer down, and then we can synthesize in 5' to 3' direction. So up here, this is now the lagging strand on that side, and here is a leading strand. So I hope you get the idea. Anyway, that's all you need to know about replication. So again, the purpose of replication is to copy the entire genome of an organism in preparation for cell division. Once the DNA has been copied, and we have two complete sets of chromosomes, then we can do cell division. Okay, so it's basically we're synthesizing the second sister chromatid to each chromosome, and then we're ready for cell division. Now, in a very simple case here, DNA replication is sort of really in a nutshell is that you have a double strand of DNA that you unwind. Each one of the original strands becomes a template strand or a parent strand, and then we're using the base pairing rules to synthesize a secondary DNA molecule. This even takes that leading lagging strand issue off here. And so the base pairing rules, it's something that you need to know. A with T and G with C. Those are the DNA base pairing rules. Of course, if you go DNA to RNA, then adenine would pair with uracil. We don't have any thymine in RNA, but in DNA we do have thymine, and so in replication we're going DNA to DNA, so A with T, G with C. And here I have the ultimate in a nutshell replication image here for you, and that is we have the two original strands in red. We call it here old strands, and then we're using base pairing rules to make the yellow, these new strands. Of course, both of the resulting DNA molecules are half old and half new, so we call that whole thing semi-conservative. And that is basically it here. So replication, both of the new strands are half old and half new, therefore the term semi-conservative, and the process is catalyzed by enzymes. DNA polymerase will do the job, which it only has one issue, that it can only go in 5' to 3' direction, but it is otherwise, it's an awesome molecule, it's an awesome enzyme, because it is extremely high fidelity, so it makes very few mistakes, and on top of making very few mistakes, it even proofreads everything and double checks that everything is right. So therefore this very low error rate. For the lagging strand, we do need DNA ligase because we need to remove these Okazaki fragments, these primer pieces, and then replace them with DNA with DNA and nucleotides, and that requires the enzyme DNA ligase. Think of DNA ligase as molecular glue. And before we go and finish this up, there's one more thing that I wanted to bring up here. I'm going back to this image right here. So because I'm going to mention that in the mitosis and cancer lecture, there's a little section on what happens, also talks about the shortening of telomeric ends and how that makes a difference for cell aging and aging in general. So if you think about here the lagging strand, and remember we always have to put down a new primer pair. So let's think about this one right here. So here we're going on our lagging strand. So here the leading strand, again, no problem. But the lagging strand, we have to go in this direction. So we just finished this section. This one was already finished. Now we have to wait for a new piece to open up. We'll start putting another primer in here, and then we'll finish up our lagging strand right here. Now the problem is there's a little piece here at the very end. This piece right here, from here to here, so from the very end of that, the 3' end of this chromosome to the last primer we were able to put down. There's a little piece that we won't be able to finish because there is no more primer. We can't put down a primer into empty space, and so we're going to lose this little piece here. And this is the reason why cells age and why they only have a limited amount of cell divisions available. And so the telomeric end problem, so we have a little buffer zone on all of our chromosomal ends, because of course this happens on the other side too. So on this chromosome, the very end, we're going to lose a little bit of information, and that's okay if it's just telomeric end sequence. But once it cuts into the coding sequences or into something important, then the cells behave abnormally. And that's a big reason why cancer cells become more and more abnormal, because they undergo multiple cell divisions. They don't want to die. They don't undergo this aging and death process that's normal when cells have undergone a certain number of cell divisions. Then they need to die because we have cut too much into these telomeric end sequences. So cancer cells, they don't care. They keep dividing, and with every replication round, they lose a little bit of their telomeric end sequences, and they start behaving abnormal and turn even more abnormal as they do more cell divisions, more and more cell divisions. So that's something that will come up again in the cancer lecture. That's sort of attached to the mitosis cell division lecture, but just to keep that in mind. So here the issue with replication is on the lagging strand, we're losing a little bit of sequence with every round of replication, and that is the reason for cell aging and cell death. Normal cells, they will die after a certain number of cell divisions, versus cancer cells, they do not. Okay, so I'm done, and I'm going to post this for you right now.